Early stress alters sperm DNA, impacting offspring's brain development. Summary: childhood stress may leave lasting marks on sperm, altering epigenetic profiles and potentially influencing brain development in offspring. Researchers found that men with high levels of childhood maltreatment had changes in DNA methylation and non-coding RNA levels in their sperm. These epigenetic changes could mediate how early life stress impacts the next generation, highlighting the potential for intergenerational effects. While the study demonstrates significant associations, further research is needed to confirm the role of epigenetic inheritance in humans. Key facts: sperm epigenetic changes, childhood maltreatment, altered DNA methylation, and non-coding RNA in sperm. Brain development links. These changes are associated with brain development in offspring. Intergenerational impact. Findings suggest early life stress may influence future generations via epigenetics. Source: University of Turku. Childhood stress can impact the epigenetic profile of sperm. These results may also have practical implications for future generations through epigenetic inheritance, as many of the observed epigenetic associations are related to brain development. This groundbreaking discovery has been made in the Fin Brain study at the University of Turku, Finland, in collaboration with researchers from the University of Copenhagen, Denmark. Epigenetic inheritance refers to the transmission of information on acquired conditions to the next generation without it being encoded in the DNA sequence. Changes caused by the environment may therefore be passed on from generation to generation through gametes. Epigenetic changes in gametes have a special role as they can also affect subsequent generations via epigenetic inheritance. Credit: Neuroscience News. Researchers from the Fin Brain study at the University of Turku have previously found an association between paternal early life stress exposure and the child's brain development. In a recent study, childhood maltreatment was found to be associated with several sperm epigenetic characteristics that may mediate the effects on offspring brain development, including expression levels of the non-coding RNA molecule AQR435P and methylation of the CRC1 and GBX2 genes. Next, we want to study childhood maltreatment, epigenome of sperm, and offspring characteristics together. Demonstrating epigenetic inheritance in humans would rewrite the rules of inheritance, which highlights the need for further research, says Jetro Tulari, who is the first author of the article. Although all cells in the body have the same genes and DNA, they have distinct epigenetic profiles, which determines different appearance and function. Epigenetic regulation thus enables differences in the cells of the brain, muscles, and skin by silencing and activating genes. Epigenetic changes in gametes have a special role, as they can also affect subsequent generations via epigenetic inheritance. There are currently several projects studying the phenomenon in both animal models and humans. This study was the largest and most comprehensive human study conducted so far, says Professor Noura Kotaja, a senior researcher in the study. The study found lower levels of sperm DNA methylation in 3 DNA regions and changed levels of several small non-coding RNA molecules in men with high levels of childhood maltreatment. They were compared with men who had few experiences of maltreatment, as measured by questionnaires. Altogether, 55 middle-aged men took part in the study. In the statistical modeling, many other health factors, such as the respondents' age, weight, and smoking, were controlled for. The Fin Brain cohort and other multi-generational data from the Center for Population Research will enable other similar high-quality studies on human epigenetic inheritance. However, the inheritance of these findings has not yet been proven, so further research is needed, says Professor Emeritus Hasse Carlson, who initiated the Fin Brain study. Launched in 2010, Fin Brain is a birth cohort study of the University of Turku, involving over 4,000 families and aiming at exploring environmental and genetic factors influencing the development of a child. The parent study of the cohort is also a key part of the project's research, and this sub-study focuses on paternal health in the cohort. About this epigenetics research news. Author Tuomas Koivula. Source University of Turku. Contact Tuomas Koivula, University of Turku. Image. The image is credited to Neuroscience News. Original research open access. Exposure to childhood maltreatment is associated with specific epigenetic patterns in sperm, by Jetro J. Tulari et al. Molecular Psychiatry. Abstract. Exposure to childhood maltreatment is associated with specific epigenetic patterns in sperm. Childhood maltreatment exposure increases the risk of adverse long-term health consequences for the exposed individual. Animal studies suggest that CME may also influence the health and behavior in the next generation, offspring through CME-driven epigenetic changes in the germline. Here we investigated the associated between early life stress on the epigenome of sperm in humans with history of CME. We measured paternal CME using the Trauma and Distress Scale questionnaire and mapped sperm-borne senRNAs, expressional RNA sequencing, and DNA methylation in spermatozoa by reduced representation bisulfite sequencing in males from the fin brain birth. Cohort study. The study design was a case control study, high TADS and low TADS. We identified 3 genomic regions with differential methylation between low and high TADS and 68 tRNA, derived small RNAs and mRNAs with different levels in males with high CME. Of potential interest, we identified differential expression of mRNA mir34C55P and differential methylation levels near the CRTC1 and GBX2 genes, which are documented to control brain development. Our results provide further evidence that early life stress influences the paternal germline epigenome and supports a possible effect in modulating the development of the central nervous system of the next generation.